Hello and welcome to Chapter 5, 1 of 5. We are going to talk about nuclear division. Okay, so cells reproduce by dividing and passing on their genes to daughter cells. The nucleus always divides before the cell divides, and each daughter cell will contain its own nucleus. So if you're looking at the diagram to the right, you see a diagram of mitosis. And in this, you see a cell with 46 chromosomes. The chromosomes align at the metaphase plate. They replicate. The nucleus splits, and the cytoplasm begins to pull apart. Those centrioles are pulling those chromosomes apart. And then in the end, you have two daughter nuclei, which are genetically identical to the parent cell. And remember, it's super important. Mitosis is asexual reproduction. Okay, you end up with daughter cells that are identical to the parent cell. So nuclear division combined with cell division allows cells to reproduce themselves and to grow. So again, asexual reproduction. All cells in your body are genetically identical except for gametes, right? Gametes are reproductive cells, so those would be examples of eggs or sperm. So all cells are derived from one cell, which is a zygote. Before division in eukaryotic cells, thread-like structures called chromosomes become visible, and the number of chromosomes depends on the species. So humans have 46. Flies have 8. So looking at this, 46 chromosomes, we know that we have 23 pairs of like chromosomes, okay, and one is from mother, one set is from your mother and one set is from your father, which is why you have 46. So if you can stain them very intensely, you can create what's called a karyogram. So here's a karyogram. It's a prepared photograph of chromosomes. And you can see pairs 1 through 23. So matching pairs are called homologous pairs. And in humans, there are 2 sets of 23 paired chromosomes, 1 set from mother and 1 set from father. So if you're looking at this picture here, you can see that set number 1 at the top left here, they're homologous, right? They look similar. They're the same height. Their sections are proportioned equally. They are homologous chromosomes. So it's the number 1 chromosome from mom and the number 1 chromosome from dad that make up that homologous pair. Autosomes are numbered 1 through 22. So that's all of these chromosomes that you're looking at here, 1 through 22, are homologous pairs. They're autosomes. They're non-sex chromosomes. The sex chromosomes are going to be number 23, and they're not actually numbered in the same way because they're not a homologous pair, right? So you have, well, you maybe not. So if you are a male, you have an X and a Y. And if you are a female, then you have an X and an X. So each chromosome has a characteristic set of genes which code for different features. And that's the other reason why they would be considered homologous pairs because, and this is just a completely theoretical example, but number 1 here might carry the genes for how your internal organs are shaped. Okay, again, completely hypothetical. So the one from your mom will determine how your organs are shaped, and the one from your dad will determine how your organs are shaped. And number 2 might be about blood cell size, right? So again, completely hypothetical, but you get the idea that these different pairs of homologous chromosomes will code for different features. Cells that contain 2 sets of chromosomes are described as diploid, right? Diploid, and are represented by 2N. So N is the number of chromosomes that are in one set. And a cell which contains only 1 set of chromosomes is described as haploid and is represented by N. So a cell that has a set from mom and a set from dad is diploid. Okay, it has 2 sets of chromosomes. A cell that only has 1 set of chromosome is haploid. Okay. And an example of that would be gametes, so sperm and eggs, right? They only have 1 set of chromosomes. So a double structure made of two identical structures called chromatids are what you see here on the right, and they're joined together in the middle by the centromere. So during interphase, which is the period between nuclear divisions, each DNA molecule makes an identical copy of itself. And excuse me, we're all used to seeing chromosomes as this like characteristic X shape. In actuality, when DNA is in free form inside the nucleus, it looks like a big pile of spaghetti. However, when it's about time to replicate, it will start condensing and coiling into just half of this, so just the yellow part. And then when it actually is in the process during interphase of replicating, it is making a copy of itself, and that's what this blue side is. So when you're looking at this sort of like X representation of a chromosome, really the yellow side is, not including mutations, the yellow side and the blue side would be identical sister chromatids. Each chromatid contains one molecule of DNA. So DNA is the molecule of inheritance that is made up of this series of genes, right? Which is one unit of inheritance. One gene codes for one polypeptide involved in a specific aspect of functioning of an organism. Okay, so like how well your liver is going to function is partially based on genetics. Okay, so the gene for a particular characteristic is always found at the same position or locus. Okay, plural would be loci. The total number of different genes in humans is thought to be 20,000 to 25,000, and this is protein coding genes. Okay, so this is why you have a homologous pair. So if you look at this homologous pair of chromosomes, you have one from mom and one from dad, right? One from the male, one from the female. And again, see, they're in an unduplicated state. They don't look like X's. Okay, so if you look at one particular locus, you'll be looking at one particular gene. Okay, and when you're looking at that one particular gene on the chromosome that you got from the female and the same gene in the same locus that you got from the male, you're looking at a homologous pair of alleles. Okay. So each member of homologous chromosomes come from one of the parents. So again, 23 from the female, 23 from the paternal, which equal 23 homologous pairs, okay? 46 chromosomes in total for humans. And each member of a homologous pair possesses genes controlling the same characteristics. Again, you're not going to only get, unless there's a faulty unless there's some kind of issue, you're not going to get just a gene for immune system from your mom, right? You get that same gene from your mom and your dad. And a gene for a different characteristic may exist in different forms, which can be expressed differently. Okay. So an example that I want to talk about is cystic fibrosis. This is a condition that is caused by a faulty allele, so a mutation of a gene that codes for a chloride channel protein. And we've talked about this before when we talked about cell surface membranes. Okay, so it's a gene that is coding for chloride channel protein, which is needed to produce mucus in the lungs and in other places in your body. And this faulty gene, this mutation, doesn't allow the chloride ions to populate as they normally would, which causes very thick mucus to accumulate. And both homologous chromosomes have to have the mutation in order to get cystic fibrosis. If only the mom has it or only the dad has it, you will not get cystic fibrosis, but you will be a carrier. Okay, nuclear division. So as a zygote matures into adult with trillions of cells, each cell must be genetically identical, and this type of nuclear division is called mitosis. And this is an example of asexual reproduction that many organisms might go through, or it's also an example of how, if you have an injury in your body, how your cells will heal itself, how they'll create new cells to heal. Sexual reproduction, on the other hand, is the point before fertilization where chromosomes must be halved to create haploid cells. So this type of nuclear division is called meiosis, or reduction division, and it creates dissimilar offspring. Okay, so not clones as in mitosis, but genetically dissimilar offspring. Okay. And so with meiosis, so we talked about how at the beginning of meiosis and mitosis, you start with a diploid cell. Okay, so you start with 46 chromosomes. And in mitosis, you get one cell cycle of division, and you end up with two identical daughter cells that are both diploid, right? They're identical to the parent diploid cell. But in meiosis, you end up with approximately four daughter cells that are dissimilar to the parent and have gone through 2 rounds of division. They are now haploid, meaning that they reduced from being a diploid cell with 46 chromosomes to a haploid cell with 23. The regular sequence of events that takes place between one cell division to the next are called phases, and there's 3 distinct phases: interphase, nuclear division, and cell division. Okay, so the G1 phase, the cell grows to the normal size after cell division. And then in the S, and this is all interphase, okay, so interphase G1, interphase S, and interphase G2. So interphase G1, cell grows to the normal size after cell division. Interphase S phase, the cell receives a signal to start dividing and begins synthesizing DNA. Interphase G2 is when DNA is checked and repaired, cell continues to grow and replicate organelles. Okay, so when you're looking at mitosis specifically in cytokinesis, M phase in mitosis is where the growth stops temporarily, and the nucleus will divide into daughter nuclei. Cytokinesis is whole cell division at this point, and it's involved in the constriction of the cytoplasm between the two nuclei. So you have the nucleus dividing in M phase during mitosis, and then the whole cell division in cytokinesis. For mitosis, you have early prophase and late prophase. So in early prophase, chromosomes will start to coil up and become visible when they're stained, and then the centriole replicates. In late prophase, the centrioles move to opposite sides of the nucleus. Nuclear envelope breaks up into a vesicle, okay, which is not visible, and the chromosomes are seen as identical chromatids. Okay, mitosis metaphase, centrioles reach opposite ends or poles, and they're organized by spindle microtubules. Chromosomes align at the equator of the spindle and attach to the centromeres. The anaphase in mitosis, chromatids move to opposite poles and they are pulled by the centromeres. And in telophase, chromatids have reached spindle and begin uncoiling, and nuclear envelope reforms. Okay, the biological significance of mitosis. So this is for growth. Okay, daughter cells have the same number of chromosomes as parent cells in our genetic clones. This allows for growth of multicellular organisms from unicellular organisms. Zygotes. Okay, it's for replacement of cells and repair of tissues. So cells are constantly dying and being replaced by new cells. And some animals, like some lizards, can regenerate entire body parts through the process of mitosis. Okay, it's asexual reproduction. It's the production of new individuals of a species by a single parent organism. And it can be from unicellular organisms or multicellular. And budding is particularly common in plants. Okay, cloning of B lymphocytes and T lymphocytes during immune response is dependent on mitosis.